my project is multi-material printing at the Micron-Scale. And so, sorry. So first of all, let's just chat about what is 3D printing. Everybody kind of hears it. Is this working? I'm not sure. Is it just connected to that? Oh, okay. I wasn't sure if it was projected. Here, it's kind of a buzzword. I've heard it a lot recently, especially with my project, but it's essentially just a procedure where you manufacture or rapidly prototype materials using plastics of sorts. And so these are just two different examples. This is direct write, so you have a laser that focuses into some resin and it creates a three-dimensional solid structure out of a liquid resin. And then this is a projection stereolithography system, or a projection 3D printing system, that does the same method, except for it doesn't focus the light using a laser. It just projects an image from a projection device, which I'll talk about in a bit. So why is 3D printing so useful? And there's definitely a plethora of reasons, and that's why it is a buzzword. First of all, there's rapid prototyping. A lot of professional uses. We have one in the ITLL that I actually use for my 3D printing device. I print parts for it, which is kind of interesting. And this is really useful for prototyping, just in case, say you want to have a structure that's metal, but you don't have the money to necessarily invest in having it soldered or just carved out of metal and have a machinist do it for you. So you can make it out of this plastic, which is significantly less expensive, and see if it actually fits in your system. And if it does, then you can, put forward an extra $2,000 or something to actually get it fabricated with using metal. So you can have the properties that you want, or different things. Or you can, for personal use. A lot of people are excited about having a $500 3D printer on their desktop. I can't really see the uses for it personally, but people are excited about it. And then there's microsystems that are interested in using this. This is a printed device that's prints the polymer, and then they go back in and do a 2D circuit on top of the structure using selective laser sintering, or you can deposit metal onto the surface. And this is a three-dimensional circuit using a similar technology, but they again can do it in 3 dimensions as opposed to just the 2. And then the region that I'm most interested in is the biological systems aspect. So you can do tissue engineering, which is, either you make a scaffold and can seed cells onto it, or you can actually print the cells themselves. And then also pharmaceutical testing, which is kind of a segue off of printing these biological systems, but they're hoping to extend this to, say print a bunch of, liver cells or kidney cells onto a substrate, and then send in some new drug treatment to see how a specific person's body will respond to a certain drug. So it's a very personalized medicine, and I think that's kind of the direction that medicine's going, but we're a long way off from this, but that's kind of the direction that it's going. So going into the specific system that I use and that I've found the most interest in application in is a projection stereolithography system that we had, that I just referenced. So we have a light source, and we shine this light source onto a digital micromirror device. And this digital micromirror device, just to give you guys a little background, I'm not sure if you're familiar with what it is, but essentially it's an array of micromirrors. This is a pin, the tip of a pin, just so you can kind of see how small they are. It's like each of these little micromirrors act as pixels. And so this is, say you're trying to write across, each of these would be on the on state, and that would send light. It would project some light in some direction, and each of these are in the off state. So you would get a cross of light, and then each of those regions would be dark. So this is just kind of a description of it. So if the mirrors are tilted in one direction, they send light away from the sample, and then if they're tilted in a different direction, they send light continuing on to the projected region. And then this is just the off state, and it's just kind of flat, just as a reference. And so continuing on, once we project from this DMD, we go through a lens. I use an objective so that I can get the resolution that I want. And then I focus it into, or this system focuses it into a resin, and they have this platform that essentially raises to get, you print 2D layers at one at a time. So it's just this additive layer manufacturing. And so you just essentially step and repeat, and you just project different images for different lengths of time to get different exposures. So then focusing on what this photocurable resin is, my project has gone in the direction of a lot of chemistry, so I thought it would be important to reference what this resin is, just because it is an important part of my project. So we start with, let's just simplify the whole system. So we're just printing a single layer, so one layer in this three-dimensional structure. And I use what is called a monomer, and it's just a single unit chemical system that I put a photoinitiator into, and that essentially, once I shine light onto it, acts as a radical, and then it hits a monomer, and then the monomer can attach to another monomer and then create this string and solid structure from a liquid structure. And that's really helpful to have a resin that can go from liquid to solid, so that you can pattern very easily. And so that's why I use this specific system. And so this is just designating what the monomer units are, and then the yellow is the photoinitiator. So we irradiate, and then as we irradiate, we, as I discussed, see these polymer chains. So these monomer units grow and grow and grow, and there's a lot of different types of monomers, but this is just a very simplified system. And so this region is remaining liquid while this region is solid. It's very similar to a lot of these lithography processes for nanotechnology that are used at CU right now. But mine is just, it's a little bit more versatile, I think, than some of the methods that are currently used. And so when we turn off our light source, we're just left with these two structures, and then we're able to remove the unpolymerized region, so the region that's still liquid, and then all you have left is this solid material, and that's what your structure ends up being. So current limitations of stereolithography, it's like, oh, if they have everything conquered, why are you so interested in it? Well, because I'm so interested in biology, it's really difficult. If you think about it, if you have this huge resin vat that is required for most stereolithography systems, and you want to put cells into it, if you know anything about biology, you know that if you try to keep cells alive for that long and that quantity, it's incredibly difficult. And if you want to use multiple materials or multiple different cell types or different chemical or biological kind of applications, then it's difficult to try and swap those two materials out, because that's what, if you're building up a three-dimensional structure, you have to be able to pattern two different materials in each layer. And if you're trying to do multiple materials, as I was saying, it's difficult to swap those if you're using cells. And then also, MEMS devices, microelectrical mechanical systems, to build this, as I discussed, you have to use multiple techniques for printing. You can't just, in one go, print the entire structure. And in this system, they had to leave little regions where they had to deposit, using a different mechanism, a very specific line of material. And so it's definitely a limitation, and it's just time consuming. So if they're trying to speed up the process, that's definitely a limitation. So what I aim to do is, or what I have done, is built a whole new system and redesigned it. So to try and, I guess, accomplish both of the two goals. So for my system, I have 3 different LED sources. I use 400 just to irradiate my sample. And then I use a DMD and project it through an optics and just objective lens. And then I focus down to a very specific sample plane, to do the same kind of projection system that we had talked about. But I use a window technique, and it's essentially acts as a non-stick window so that it won't polymerize. And then I'm able to move my sample stage forward and get a very defined thickness. And the thickness of each of my layers is completely dependent on the actuator that I'm using. So if my actuator has Micron-Scale accuracy, which it has right now, then I can get micron-thin layers, which is really great, especially for biological systems, which don't need more than, cells are on the order of 10 microns, so you don't need phenomenal resolution for biological systems, at least as we know of yet. And so what I've done is, instead of having this massive vat of material, I have a syringe pump that I'm able to just deposit a small amount of material into the region that I'm interested in, which would just be that little sample plane. and then I can polymerize a very small region, and then I can flush out the new material, and then repeat the process with a different monomer or a different cell if I'd like. This is obviously still a work in progress that hasn't been completely shown, but that's just, that's the end goal kind of application. So I've implemented this syringe pump, essentially just modifying it so that I can have a smaller volume and more efficiently replace the material. And then also, I am developing with one of our chemists a metallizable polymer. So what we're able to do is ultimately print this material that is able to utilize a specific chemistry of a thiol and an ene group, and have just some extra reactive groups floating off of the top of this monomer surface, and later come back and metallize the surface, which essentially just involves like putting this finished part into a solution, and then it will electrolytically plate onto the material. And we're still working on that, but we're getting really promising results, which is exciting in terms of conductive properties, and we're using copper right now. So that's one of the cool applications that I'm hoping to use it for. So I guess kind of all in all, the applications that I want my system to have are to be able to fabricate multi-material biological structures with both chemical and different mechanical, properties. So that, for instance, in this situation, this is the bone-cartilage interface, and I'm working with two different groups, the Ferguson group in mechanical engineering and the Bryant group in bioengineering, to reproduce this. But this is subchondral bone, so this is a really high modulus. It's really, really hard, and it has a lot of calcium in it. And then this is the zone of calcified cartilage, and that's a little bit. It's a less stiff modulus, but there's also it's difficult to see in this image, but there's cartilage fibers that are very stiff that are coming through this interface, and no one as of yet has been able to replicate that in a very biologically meaningful way. And that's I guess I would say one of the holy grails in orthopedic medicine, because cartilage is a really difficult biological system to replicate in the body. I've had hip surgery, so I know. So this would be a really interesting structure to try and replicate using my system, and that's what we're intending to use it for. And then also implementing this metallizable material into biological structures as well. So a member of our group, Anna, who's in the audience, is using it to, she's being like essentially using optical trapping to create muscle fibers, and if we're able to trigger those muscle fibers using metal, like say that you place metal underneath this muscle fiber, you could stimulate it maybe as it's growing, or just as a testing mechanism to see how strong the muscle fiber is. So those are two really interesting applications that I'm really interested in, but there's obviously a lot of applications for 3D printing. So in conclusion, I just would like to acknowledge and thank my advisor, Michael Cole, who is the chemist in our group, all of my group members, both Jake and Anna, who are my members in the audience, and Stephanie Bryant's group, and the Ferguson group, and then also obviously COSI and the NSF. So thank you. Yes, Dennis.
I understand how you can print like using polymers or photoinitiators, but how would you print cells?
It's a great question. So what the app, I guess, intend to use it for, and Anna's been doing this work as well. You have a hydrogel, so we use a thiol-ene chemistry that I was discussing for the metals, but it's a hydrogel because you put a lot of water into it. So you have this monomer system, and then you put cells into the monomer system so that it essentially polymerizes around each of the cells. So the cells don't actually get polymerized to anything. It's just it's the matrix around it. And then there's a lot of materials that are being developed right now that the cells can kind of eat through and connect to one another, so they're biodegradable. There's just so many different techniques, but right now we're concentrating on a hydrogel that can have a low and high modulus that we can seed cells with. Yeah.
Do you measure the resolution?
We've measured my system, so using a very well-established resin that's in the CNL, I have gotten pixel resolution, which is in my system right now 3 microns. But that's saying that my system can do that, but it's really difficult to know what the actual materials will be able to do. Like, who knows? I am still yet to really push the limits on these hydrogels, especially, and I know that there are some issues that are encountered with that, definitely, especially because it's a hydrogel and the monomer diffuses really quickly, so it can change the resolution. Any other questions? Thank you.
All right, so hi everyone, I'm Fiedler-Shanblatt. I work in the Kapteyn-Murnane group over in Gilla, and today I'm going to tell you a little bit about coherent diffractive imaging on a tabletop using an extreme ultraviolet high harmonic generation source. So first I want to motivate why we do microscopy with extreme ultraviolet or EUV light in the first place. Extreme ultraviolet and soft X-ray light has a wavelength on the order of 10 to 100 nanometers, which makes this type of light really great for imaging objects on the nanoscale, because as you know, smaller wavelengths can resolve smaller features. Furthermore, this kind of light can penetrate thick objects in certain cases, and as I said, it's capable of imaging small features. And it also has elemental and chemical specificity due to the fact that certain materials have absorption edges in the EUV. So EUV is good for high spatial resolution, but what about temporal resolution? Well, there are many different imaging techniques that are appropriate for different kinds of samples. Some techniques, such as electron microscopy and certain super resolution techniques, can get down to 1s or 10s of nanometers spatially, but temporally they can maybe only get down to milliseconds or seconds. X-ray microscopy using ultrafast pulses, on the other hand, can access nanometer length scales, as well as it has the potential to resolve time dynamics on the order of femtoseconds. Now, how do we image these objects? Well, in a traditional imaging system, an image is formed using a lens. But if you want to image something on the nanoscale using a lens, you'll run into a problem, which is that you can't make a traditional lens for EUV the way that you make, say, a glass lens for visible light. And this is because there isn't really a feasible material that has a high enough index of refraction in the EUV in order to focus the light. So there are Fresnel zone plates that are used sometimes, but they can be lossy. They're not necessarily very efficient. And furthermore, the resolution of the image that you get from them is ultimately limited by the fabrication quality of the zone plate. So this means that they can't provide diffraction-limited resolution. So another solution, which is what we do, is to do lensless coherent diffractive imaging, or CDI. This means getting rid of the lens altogether and remembering that if you propagate an image into the far field, you obtain the Fourier transform. So this is just a demonstration showing that if you start with an object, propagate it into the far field diffraction pattern looks basically identical to the Fourier transform. So after we get the Fourier transform, we reconstruct the image using a phase retrieval algorithm, which I'll tell you about in a few slides. So right now I'd like to back up for a minute and explain where the EUV light comes from in the first place. It's produced in a process called high harmonic generation, which is an extreme nonlinear process where light from a femtosecond driving laser is upconverted to EUV. Basically, the intense laser field ionizes an atom and then slams the electron back down into the parent ion, and this process releases a photon. So a wide range of wavelengths will be produced, but we can select out the wavelength that we want by using narrow bandwidth multi-layer mirrors. Furthermore, we do this process in a waveguide, and the waveguide is filled with a noble gas. So by tuning the pressure of the gas, we can phase match the phase velocity of the IR driving laser with the high harmonics. And this phase matching creates coherent addition of the high harmonic beam. So at the end, we get out a bright laser-like beam that has full spatial coherence. Before I describe the phase retrieval algorithm, I want to motivate why we bother to retrieve the phase in the first place. It turns out that the phase of the Fourier transform has a really profound influence on the amplitude of the object. So this is a demonstration where you take the Fourier transform of each of these images and then swap the intensity of one with the phase of the other in Fourier space, and then transform it back to see what you get out for your image. So you can see that the phase more strongly influences what the image looks like than the amplitude. So it's important that we want to get out the phase in the Fourier plane so that we can properly reconstruct the image. Okay, so now what kind of samples can we look at, and how do we actually get the phase? Well, first of all, in order to reconstruct anything, we need to make sure that the diffraction pattern is oversampled. This means that the intensity is being sampled at twice the Nyquist frequency, which in the image plane corresponds to the image taking up a quarter of the total area of the field of view. So basically what we do is we start with our measured amplitude, which we know because we have a CCD in the far field, and we Fourier transform it to image space, and then we apply the Nyquist constraint, which corresponds to setting 3/4 of the image equal to 0, and then Fourier transforming back to Fourier space. And so now we have some possibly goofy amplitude and some phase, right? So we keep the phase because that's what we eventually want to retrieve, but we replace the wrong amplitude with the correct amplitude that we know because we measured. And then this loop repeats, and eventually, hopefully it will converge, which means that we start getting the correct amplitude out at this stage, and we can compare it to the known amplitude. And then when that converges, we know that we've retrieved the phase. So here are some experimental results from a few years ago when CDI was done in the group using an isolated sample. And we were able to push the resolution limit by achieving a record tabletop resolution of 22 nanometers using a 13 nanometer high harmonic generation source. So this shows a typical setup for CDI. The IR beam is being focused into the waveguide, and then out of the waveguide comes the high harmonic light as well as the IR. And it bounces off a couple multi-layer mirrors, which selects out the wavelength that we want of the high harmonic beam. And then that hits the sample and it diffracts onto the CCD. So here is a SEM image of the sample and the diffraction pattern that was collected, and finally the reconstruction. In the past 5 years or so, a technique known as keyhole CDI has come out, and it allows an extended, non-isolated sample to be used by demanding that the beam be isolated rather than that the sample be isolated. And this can be achieved pretty simply by using a pinhole. So here's the schematic for that. It's similar to the previous one, just with the addition of the pinhole. And this is an optical microscope image of the sample that was used. And you can see two regions of interest. Regions 1 and 2 are circled. And down here we have two diffraction patterns that correspond to these two regions, and then two reconstructions that correspond to the regions. So recently, we also found that CDI can be used to obtain depth information from a sample. If the light is incident on a semi-transparent sample, holography can be done by using the unscattered portion of the beam, which acts as a reference wave, with the scattered portion of the beam. And so here is the schematic of the setup. You might notice that there's the addition of the second pinhole. And the purpose of the second pinhole is to reduce some scatter from the first pinhole. So it basically acts as a low-pass spatial filter. And the sample that was used for this experiment is chromium on silicon nitride with some holes punched through it and some shapes. So to test how well this worked, we compared it against AFM. So here you can see an AFM image of the sample showing the depths as well as a CDI image, and then a line out that's taken across the sample. And you can see that AFM and CDI agree pretty well within their error bars. And then down here are 3D profiles of the sample based on the depth values. And these two colors are just showing that it's 30 nanometers of chromium on top of 45 nanometers of silicon nitride. So now all of the experiments that I've been telling you about so far were done in what's called transmission mode, which is where the light passes through some part of the sample. But sometimes we want to image a sample that is very thick or that is not transparent to EUV. For example, patterned wafers that are commonly used in the semiconductor industry aren't necessarily transmissive to EUV. So if we want to image something like this, we can put it in a reflection geometry, which is where the sample is placed out, for instance, 45 degrees, and the light is reflected off of it rather than being transmitted. So before we did this using EUV, we did it in kind of a test bed experiment using a helium neon laser. So you can see the setup for that, where the sample's at 45 degrees, and then the diffraction pattern is collected. Here's an optical microscope image of the sample and some reconstructions. So you might notice, if you think about reflection geometry, that you're going to get some distortion in the horizontal direction due to the geometry of the setup. So that's seen in the diffraction pattern here, where the spacing of the peaks on the left is different from the spacing of the peaks on the right. So to correct this, we do what's called tilted plane correction, which is where we transform the diffraction pattern so that it looks like it was sampled on a linear grid. And this allows us to pass the diffraction pattern through the regular phase retrieval algorithm so that we can use Fourier transforms. So it's just a convenient way to be able to do the phase retrieval in the usual way. We've just started taking data and doing reconstructions with EUV in reflection mode. So here's some pretty new results from the past couple weeks. This shows an optical microscope image of the sample, which is 30 nanometers of titanium patterned on silicon wafer. And this is showing the reconstruction of the complex amplitude, where the intensity corresponds to the amplitude and the color corresponds to the phase. And here's just a schematic of the setup. It's pretty simple, actually. It passes through a pinhole, reflects off of mirrors, hits the sample, and then is collected. Also, we took this data using a technique known as tychography, which is where the sample is scanned across the beam. And this gives us some redundant information, which helps in the reconstructions.
How do you sample that? How do you scan it?
So just like a certain, you move the sample like certain steps that you determine, and then you have some overlap. Like if you imagine the beam as a circle, you have some overlap. Both. So it's, it's like maybe like a raster. All right, so in the future, we will continue to work on reflection mode with EUV, and we also plan on imaging new diverse types of samples, such as magnetic domains, acoustic waves, biological samples, and working towards high-resolution 3D imaging with time-resolved dynamics. I want to thank the PIs, Margaret and Henry, as well as the imaging team, Matt, Bocheng, Dennis, and Dan, and the rest of the KM group. And also thank you to COSI and others for funding, and thank you to all of our collaborators. And finally, thank all of you for your attention, and I'm happy to take any questions. Yep. Mm-hmm. Oh, I suppose you mean like why isn't the 5 on the left exactly one color?
Yeah, exactly. Like why? Yeah.
I guess there's just some error in the reconstruction. It's relative to the background, it's still quite close, but it's not completely perfect, I guess. So for one thing, obviously it's diffraction limited, but also, so one thing that I think is maybe an issue is the noise in the system. So you can imagine if you're sampling with your pixels on your detector and you have a little bit of vibration, that could cause cause like some feature to move onto a different pixel. So that can, translate into, not perfect spatial resolution, like not perfect λ/2.
When you say fraction limited, in this, since you're not using lenses, in this case, is that just determined by the collection or the size of your detector array? Or is it just from the,
So I suppose that would limit it, but it's also, yeah, I mean, it's limited, by experimental error, but I think that even with a perfect, if everything was perfectly stable and didn't move at all, and you had some image that could be, way oversampled by like your pixel density, there's still, I think, the fundamental limit of λ/2 in this case.
He's just asking what the numerical aperture.
Oh. Well, I guess the numerical aperture depends on like which experiment, right? I don't know the numbers off the top of my head, but in general, all of these are very high numerical aperture compared to some other techniques.
What kind of image are you using for this pixels?
So we use so that depends exactly on the experiment, but we use an Andor CCD, which has, I believe the pixels are 13.5 microns, and it has about, is it 2,000 pixels? 2,000 by 2,000. And the exact distance of the sample varies. It partially has to do with how we align it, but it's on the order of, I mean, for reflection mode, I believe the distance is on the order of maybe 6 centimeters.
no more questions, then let's maybe listen it again.
Oh, yeah.
You mentioned the time resolution, potential time resolution. What is it?